Hola a todos y a todas, gracias por seguir con este evento para Latinoamérica. Mi nombre es Angie Díaz, soy miembro de la Sociedad Chilena de Evolución y estaré a cargo en este instante de presentarles a Sergio Nigenda-Morales, quien nos expondrá a continuación. Sergio es investigador postdoctoral del Centro de Investigaciones y Estudios Avanzados de México, donde estudia cómo los cambios demográficos y ambientales impactan el potencial adaptativo de poblaciones silvestres de mamíferos. Hoy, Sergio nos hablará sobre los efectos genómicos de la caza de ballenas y el aislamiento en poblaciones de rorcual común.
Yes, I'm here. I'm sorry. Yes, I'm already sharing my screen. Can you see my screen now? I think you do. Can you see it? I think you do. Okay. Okay, excellent. Thank you very much, Angie, for your very kind introduction. As you stated, and I would also like to thank you all for being here today for my presentation, where I'm going to talk about the genomic effects for the whale hunting and the isolation effect in different populations. And one of the most common questions in this evolutionary genetics is how different natural populations lose their genetic variability and what different genetical aspects are being considered whenever we talk about this specific phenomena, and the different consequences in the long term of these losses that we may have for the fitness or for the well-being or the adequation of these populations. So one of the ways, or the most common way in which there is going to be a genetic loss, is by changes in the size or some diminishment in the population size, also commonly known as bottlenecks, a populational bottlenecks. And depending on the magnitude and the lasting of these bottlenecks that may be caused, either caused by natural ways or natural phenomena or anthropogenic reasons, we may have these bottlenecks, or they may impact the genetical variation, as we can see in this graph, where you can see that after a bottleneck, there was a reduction of 97%. So here in yellow, you can see that whenever we don't have migration, there is going to be a specific variation, a specific diminishment in the genetical variation. And here we can see how it is not going to be impacted whenever we have immigration. But if we have any, let's say, any variation, then we're going to see a variation as well. Another consequence of these bottlenecks is the endogamic patterns or behavior that we can see in different populations. And this is also going to increase the genic load, which is pretty much going to impact the genetical fitness of a population in comparison to some other species that, or let's say in comparison to some others that could be specifically measured as the accumulation of these variations. So that means that it is going to have a specific harmful consequence for future generations. So here we can see in yellow that this genetic load after bottleneck increases whenever we have immigration towards the population, and whenever we have immigration, we are going to see how this is going to happen. And so here we can see that this is some specific variation. And that's why it is important to determine how long or when these bottlenecks occur or happen, and also how long they last, as to see how they are going to recover or whether they are going to become extinct. However, being able to calculate these different populational density and long-lasting effects of these bottlenecks, it is very complex given the specific genetical data, because this genetical data is giving us, let's say, some specific information that dates back, let's say, long time back. So this is telling us, so this is so it is very difficult to tell information or to be able to assess what happened in time if it is, let's say, very recent in time. However, thanks to technology and thanks to the genomic sequencing that we have, we know that this might be useful to determine these parameters whenever we have seen some specific reductions in time. And whenever we talk about this whaling problem that we have, this whaling and how they have made some whales to become extinct. And amongst those species, we have the species that was highly impacted by this rorqual commune at the North Pacific. They hunted more than 1,000 of these animals, and or let's say they were hunted in of the oceans, but here we can see that the lowest numbers they got around 1960 and 1980, and the estimation of this impact of this whale hunting, it is still difficult to assess how, let's say, how deep the impact was, because sometimes it is estimated that they were not as hunted as much as it is believed. However, here we can see, and as I was saying, there are some specific information that it's not very easy to read, given the genetic information that we can extract from them. Another population, or besides the North Pacific, there's another population that lives in the California Gulf, in the Gulf of California and down here to Mexico. And this population lives here and has been isolated for thousands of years. And there are some specific reasons for having such a situation. And it is estimated that here we have more than between 300 and 600 individuals, thanks to different data that we have collected. And we know that they have never been hunted. And that's why they have been isolated. So what we want to do, and this is the Gulf of California, and here we have the different satellites landmarks where we have, they were appointed. And as you can see, none of these went to the Pacific. So they are going to be there all year long in this Gulf of California, and they don't do any migration. So we wanted to assess what would be the specific effects of these isolated populations that were, let's say, that didn't have any impact by anthropogenic reasons. And another one, we wanted to assess those impacts between these two groups, the ones that were impacted by the anthropogenic reasons and the ones that were affected by the natural reasons. So we worked with a population of 30 individuals in the Northwestern Pacific, and then a population of 30 in the Gulf of California, and we have a coverage of 27 times. So this is what we have between these two different areas. And the genome of reference was the, because I'm sorry, I forgot to say this, but this is usually well known as the mink whale. So we wanted to have a different species, the fin whale, but we had to work with the mink or mink whale. So the results that we have is that on the left side, we have all of the individuals for the Northwestern Pacific. And here we can see a broader variety in genetic terms. And the ones that are in the Gulf of California, they have a lower heterozygosity. So when we see this in a different way or different perspective of this, whenever we start to study these genomes in different lines, we can see the different lines. So here we can see 1 million of base pairs, and we can see a lower degree of this in the ones that were isolated. But in this histogram, it is possible to see how it is a greater enriching of different windows of, let's say, very low heterozygosity from 0 to 400, which is not the exact case for the specific cases in the Pacific Northwestern area. After that, we worked with the different traces of homozygotes, or how they're called, ROH, which are specific homozygotes. So there is no great variation, and they have a common ancestor that is, let's say, very recent, relatively recent. And here we can see how we categorize them between amongst short, intermediate, and long these traces. So the short traces are telling us about old endogamy, and the long ones are telling us about a recent endogamy. So here we have all of this different information coming from the Northwestern Pacific area, and there are very low, or there is very low variation in their genomic line. And in terms of this homozygosity, so here we have only 5% of variation, whereas in the Gulf of California, we have 38% of variation. But in both cases, these traces of homozygosity, or these ROAs, are shorts. So this is telling us that in both cases, there is not very recent endogamy or endogamic relationships. And here we have also in the Gulf of California, we have longer traces, and this is telling us that we have different behaviors. So it is possible to conclude in terms of genetic diversity that besides, or let's say, even though these animals were hunted in the past, they have a broader diversity in comparison to the Gulf of California, where we have a specific, where we have the specific demographics that have traditionally been limited. What we also saw in some literature review, together with Paulina Núñez, we conducted different demographic models, and we could see one that is very well adjusted to the data we had, and that has to do with the model that has two different changes in size. And let's remember that these are effective sizes over the population, so they are not under census. So under 16,000 effective in 1,000 years, and the next change in size was 23 to 300 individuals, which is a lot. So this represents more than 99%. So this was seen just 2 generations ago. And let's say with the generation, having in mind this generational time, this could represent, let's say, 26 years. So this diminishment was given 2 generations ago, and this responds to 1954 and 1980, if we have in mind that the samples were collected around 2006 for this study. So these samples were collected back in 2006 for this study. So that's why it is possible to determine were there, or let's say how old these samples were. So this is telling us that it is possible to conduct an estimation, and let's say very reliable, it is possible to raise very reliable hypothesis with the collection of data that we can conduct today. So on top of that, we can also see the changes or the expansions that we could see in the other model, and this happened in the last glacier that we have 1,600 years ago, and in the Gulf of California, it has remained pretty much the same. So here we have about 100 individuals, and there is very low migration towards the Gulf of California. So there is almost 1 migrant per generation, and there is a greater migration the other way around, outwards towards the Pacific. So this is telling us that the pattern is that this area has been isolated for longer, and this would tell us that this has happened for the last 1,600 years. So here we can see how the proteins can also affect, and how this can be classified in synonyms if it is not affecting proteins, and also if it has a harmful effect, and also in the loss of functions, whether the protein lost its function, so those are the deleterious, if to call them so. And we could also see that in the Gulf of California, there is an increase in the homozygosity in all of the different categories. So synonyms, deleterious, and loss of function. So there is a specific accumulation of these deleterious, and depending on the function they have in terms of or regarding the Northwestern Pacific area. So with all of the different categories, but especially it is especially important for the losses function, which is specifically important for the Gulf of California, and this is pretty much affecting the fitness of this population. And this analysis was conducted by Mei-Shi Liu, who's a PhD student. And lastly, we given that we don't know, because the very little that we know is that there is a short migration, or let's say a very slight migration towards the Pacific area. So we could tell that this is what happened, or at least that's the information that we have from our model. So we started to work with different modeling with 40 genomic species, and to study this migration as to see what would happen if there were no migration, because with these simulations that we conducted, were aimed to do so. So what we saw with these simulations is that there could be, let's say, values very close to what we actually got. But if there weren't any migration, which is this last bar, then the genetic load would be even higher. So there would be, let's say, more accumulation of the different aspects that we assess with the California group. And on the other hand, we saw that if there weren't any migration, this heterozygosity could be almost very close to zero. So what these simulations are telling us is that this genomic flow is key as to maintain the sustainability of these species in the long term. And some of the conclusions that we could raise is that the whale hunting impacted or dramatically impacted the effective size of the population in the Northwestern Pacific. And even though we had that situation, it was possible to maintain or to keep this genetic variation, because this bottleneck didn't last too much, only 2 generations. So that's why we didn't see any genetic loss. And it is also possible to estimate the time of these genetic losses with some specific genomic studies. And this is also, let's say, there are also some other subtle considerations to have in mind, because it depends very much on the species and also on the biology of this species. And we can also see how there was a specific species that was isolated in the Gulf of California, and their behavior pretty much proves what we have studied. And the low genomic flow, which is just less than or one individual migrating per generation. So this is telling us that with less than one, it is possible to accumulate some genomic load, but this is going to contribute to the survival of these animals and this is going to impact this species in the long term, especially for everything that we have seen in the Gulf of California. And also having in mind what we have here in this Gulf, it is also important to keep on studying what's going on here, because we have to have some specific protected control, but we could also endanger this species, and the recovery of this population in the Pacific area must be updated in order to recover some species, because we see that the impact was also very important. So with that, I would conclude, and I would like to thank all of the different organizations that sponsored this research, as well as my collaborators coming from different countries, such as the United States, Mexico, and Chile, and others. Sergio, I'm sorry, we have to interrupt your presentation. We're running out of time. Yes, thank you very much. And if you have any questions, just let me know. Sergio, thank you very much for your very interesting talk. Unfortunately, we don't have any time left to run a Q&A, but this is just an invitation for you to keep an eye open for the Q&A sections, so you can answer those questions there.